dilemma.
Okay. So the first part of the cell cycle is gap 1. In gap 1, the cell grows in size for a little bit. And what happens also is the organelles in the cell is duplicated. So there are 2 mitochondria here and there's some Golgi bodies here. So the organelles are duplicated. And there's loose DNA in the cell. So what is this called? It's called chromatins. Next, we move on to synthesis. What happens in synthesis is DNA synthesis. DNA synthesis is when the DNA, or in this case, chromatins, are duplicated. So in the cell, there are 2 chromatins, and there are still 2 organelles from the previous stage. Then we have gap 2. In gap 2, all there is additional growth in the cell. Gap 1, synthesis, and gap 2 are all part of interphase.
In prophase, there is 2 centrioles which start to move to opposite sides with the spindle fibers forming, and then the DNA gets tightly coiled by histones to form chromosomes, and the nuclear membrane starts to kind of deform.
Then, in metaphase, the spindle fibers attach to the centriole of each chromosome, and the centrioles start to move to opposite sides of the cell. Oh, and they line the chromosomes along the cell equator.
In anaphase, the chromatomes break apart into sister chromatids. The spindle fibers start to get shorter, and then it starts to kind of get ready to separate.
In telophase, the nucleus starts to spread apart and split into 2, and the sister chromatids go into each side, and the nuclear membrane starts to form.
And all of these cycles are part of my process. Then in cytokinesis, the nuclear membrane forms, the furrow cleavage splits the cell in half, and so now you have 2 in animal cells. And then in plant cells, it's the plant wall, or plant plate. And then you have 2 sister cells which are identical to the parent cell.
All over again.